Okay, here it is. Okay.
So maybe while the speaker is setting up the presentation, we start. Hello everyone. I'm Babek Faryabi, Assistant Professor of Pathology and a member of Penn Epigenetics Institute and Abramson Cancer Center. It's my great pleasure to introduce Dr. Leonid Mirny, who has kindly joined us from Boston today. Dr. Mirny received his PhD in biophysics from Harvard University, and after a few years as a junior fellow at the Harvard Society of Fellows, he joined the faculty of MIT in 2001, where he's currently a professor of medical engineering and science, a professor of physics, and the founding co-director of the Center for 3D Structure and Physics of the Genome. Dr. Mirny's research has truly revolutionized how we think about the principles of genome folding. In one of his many seminal works, Dr. Mirny showed that the folding of genome follows fractal globule polymer configuration, which is basically beautifully explains why chromatin can pack in such a small space and at the same time being able to dynamically fold and unfold. More recently, Dr. Mirny introduced the concept of loop extrusion that allowed us to better understand long-range interaction in the genome, and we will hear more about this topic today. In addition to these fundamental biological discoveries, Dr. Mirny's team have been developing scalable algorithms and data structures that are standard in the field, and I'm sure many of you routinely use in your own research. Dr. Mirny is a fellow of American Physical Society. In addition to that, he has been recognized by many additional awards. One of them I found very interesting is the Web of Science top 1% highly cited researcher in the last decade. So without further ado, please join me to welcome Dr. Mirny for our today's seminar that is jointly sponsored by Institute for Biomedical Informatics and Penn Epigenetics Institute. Thank you all.
Okay, thank you for inviting me. Let me share my screen. It's been a great pleasure meeting with all the people at Penn today. I don't know what's the best way of presenting from Keynote. At times it works. No, not this time. Because you would see this slide, but you wouldn't see me changing slides. I would have to share the whole screen. Give me one sec. It's a bit unpredictable what works and what doesn't. And this way you should be able to see my presentation. Okay, excellent. And I hope I can see some of you. Okay, so I'll tell you about our recent work. So I decided to kind of give a shorter seminar. It's like a half an hour seminar, maybe 35 minutes, such that we can have time for questions, and I will be stopping for questions at a few places through the presentation, because it's really hard to stay focused for a long period of time over Zoom. Okay, so chromosome conformation capture data, you certainly, I'm pretty sure most of the people on the seminar are familiar with. This is essentially an advertisement for the browser that Niels Gellenborg's lab at Harvard developed, again, with our participation. It's a really, really cool browser that allows you to browse high-C data the way you browse Google Maps. But the reason I'm playing this presentation is to highlight the idea that there are structures in high-C maps at all scales. And this is really striking. I don't know, kind of when I look at this myself, the whole notion that you can see things at the scale of the whole chromosome, whole genome, and at the same time see things that are at the scale of continents and countries and regions is really, I don't know, kind of really interesting and surprising. So it's a very multi-layered organization. And we're typically focusing on things that are very local, because that's where lots of interesting stuff is going on. And just to make sure that we're all on the same page, I will quickly remind everybody what are the main two levels or layers of organization that we and others have been analyzing over the years. So if I take a fragment of a high-C map, the two most obvious features are the AB compartments, so the checkerboards on the map, and they largely reflect spatial segregation of active chromatin that we typically refer as A chromatin and inactive as B. And obviously it's an oversimplification to say that there are two types of regions. There might be presumably gradation of heterochromatin, gradation of euchromatin, but we generally see that inactive regions are spatially separated from active ones, and that's why we see kind of depletion of A-B contacts. At the same time, there are features that people collectively call TADs. The reason they're called TADs, or topologically associating domains, is that on high-C maps of 2013, basically 7 years ago or 8 years ago, all these features were seen as triangles next to diagonals. Nowadays, we see them as very different features, because now we see all the stripes, all the stripes and dots on these maps that were not seen earlier. So we call them TADs and TAD-associated features, and I will be talking more about them through my presentation. But an important take-home message from this slide is that there are two layers of organization, and what we've shown with Francois Spitz is that you can perturb them individually. At least there was no good way of perturbing compartments, but you can essentially erase all the TADs and TAD-associated features, and compartments stay unchanged. So that's why to the first approximation, we don't need to worry about the interplay between these levels, but this is something, again, this interplay is something that I'm sure would be something very interesting to explore in the incoming years. And so that's what my group is particularly interested in. So this is just a recap of what I showed you. So that there are checkerboard interactions, and these are compartments. And then there are smaller features like TADs and dots and stripes close to diagonal. Another thing that I want to emphasize is that if you look at this data in different cell types, these data sets can look vastly different. And that's a useful thing. Again, you can just browse this data on the 4DN browser. So these are in fact micro-C data for ES cells on the left and fibroblasts on the right. And you see that in this particular region, and I literally browse through these maps kind of in synchrony. I move the left and the right one, just see what's there. In ES cells, compartments are weaker and compartments are stronger in fibroblasts. In another region, it's the other way. Kind of, I see some features, some organization in fibroblasts that are more visible than, I see some features in ES cells, and in fibroblasts, they seem to be rather big, rather boring region. So, and then I can keep just browsing. So things are different between different cell types. And again, in many cases, these differences are reflected at the level of interplay between compartments and this near diagonal features. For high-C aficionados, I will show this really amazing data set. So it's a bioarchive that you can find. So these are data from bacteria, and these are high-C maps that are absolutely impossible to even imagine what is the folding of chromatin there. So these are some amazing high-C maps from Zindan Wang's lab at the University of Indiana. And so again, these are engineered bacterial strains, and they're engineered such that loop extruders are loaded at various places in the genome. And as a result of interactions between these loop extruders, in this case, actually bypassing of them, so they can meet and bypass, you get all these crazy patterns. So if you're into crazy high C, you should absolutely check out this bioarchive. But coming back to more kind of mammalian high C, so we see these TED-associated features, and to explain the origin of these features, what we've done in 2015, we were looking for mechanisms using polymer simulations. Essentially, what I can do as a theoretician, I can't really tell you how things work, but I can tell you how things cannot work. And I get some questions in the chat, so let me see what's there. Okay, please type your questions in the chat. Okay, good. Please type your questions in the chat or raise your hand in Zoom. I'm going to see this. So as theoreticians, we can just kind of think of possible mechanisms and see what's possible out there, and then rule out lots of things. So we were looking for a mechanism to explain formation of TEDs and all these associated features, dots and stripes. And we realized that lots of mechanisms that people have proposed in the literature would not work. For example, just having a linker, kind of long and stiff piece of DNA at the boundary would not produce TEDs. Having a flexible, super flexible region would not produce TEDs, and so on. And the mechanism that did produce them was a mechanism that at that moment we were exploring with John Marco as a mechanism of genome compaction. And John referred to it as loop extrusion. And the idea is that there's going to be a hypothetical motor that lands on DNA and forms a progressively large loop. So then when two loop extruders meet, they would block each other. And then this loop extruder should fall off the DNA. So that was our model for mitotic compaction. But then we say, okay, what if, in addition to this, there is also a boundary element? And once we have this boundary element, we might be able kind of I will be using another screen to annotate things if I need to. So essentially, if I have this boundary element, so then one loop extruder comes to the boundary element, one of the motors gets stopped, but another motor continues extruding DNA. This way, so we hope that would explain how TEDs and other features are being produced. And indeed, what simulations have shown us, so we simulate a large polymer subject to loop extrusion and boundaries. And what we did observe are these interesting high C maps with domains that look like TEDs. We were actually unhappy about the way they looked, because it was 2015 and 14, and at that moment, as I told you, high C maps look, so TEDs look like rather uniform triangles next to diagonal. So we were actually surprised that there are the stripes, and there are these dots. So we were kind of disappointed, and that continued until the high resolution data came out from Erez Lieberman's lab, which indeed showed very nice stripes and dots and things that we saw in simulations, but were kind of reluctant to publish and analyze, primarily because we didn't see them in high C data. Certainly high resolution high C kind of supported our hypothesis that extrusion is underlying all of these features. So one important conceptual thing that came from this 2015 and 16 story is that what extrusion does, it facilitates all contacts. It does not facilitate contact selectively. So if you compare, for example, a map without extrusion and a map with extrusion, all contacts are facilitated. All distances are smaller, at least at the scale of extruded loops. And again, the scales of extruded loops, so this scales, this is roughly hundreds of kilobases. All these interactions are facilitated, and there are, but you do not get any sort of features that are visible on the population average map. So to get these features visible, you need boundaries. Once you establish boundaries, then you essentially eliminate certain positions for loop extruders, and that would enrich interactions within domains that would create stripes because all the interactions that are lost between TADs are now accumulated at the boundaries. And you would get these dots. Again, these are interactions that are otherwise would be depleted between the two domains. So they kind of accumulate between the two boundaries. So conceptually, all these interactions are facilitated by extrusion, but some are facilitated more than others. So another important thing that we learn from this, and so that comes back to many misconceptions or misleading illustrations that you see in the literature, is that if you create a fixed loop, so what we've done here, we essentially make these two regions stop extrusion, but we do not really facilitate lots of loading of cohesion in between. So essentially, this creates a fixed loop, like many drawings in the papers. And what we see in simulations is that once you have these fixed loops, there is no enrichment of interactions within a domain compared to between domains. So there are essentially no TADs, if you wish. You certainly do get a corner peak. That's good. But there is no insulation or facilitation to an extent, or selective facilitation. So all these papers that claim that two neighboring loops are kind of isolated from each other, that's not quite true, because this neighboring loop is not really insulated from the one next to it. And there is very little facilitation of stuff within loops. So there are many misleading illustrations that argue that this is a fixed loop. So the correct way to think why you get this facilitation, and that's where models are incredibly useful, is that you get this facilitation if you continuously load cohesion. So it's a perpetual motion, loading and unloading of cohesion. And again, nowadays we know that this loop extruder is cohesion, and the characteristic time over which this loading is happening is about 10 minutes. So there were beautiful experiments where people measured cohesion turnover time by Daniel Gerlich and Anders Hansen. And so this roughly gives you 5 to 20 minutes of turnover time. So as long as this is happening, cohesion loads creates kind of these tracks in individual cells. And this tracks in individual cells kind of fill the interior of this, of a TAD. So that's the basic idea. So these are not fixed loops. So fixed loops provide no facilitation within a loop and no insulation of neighboring loops from each other. So I will stop for questions maybe after the next slide. So what evidence do we have for this mechanism actually operating? The strongest evidence come from depletion experiments. And in these depletion experiments, it's either cohesion being depleted or CTCF being depleted, or both. And you know, in these experiments, what people have seen is that you essentially see, so on the right, you would see enrichment of cohesion. On the left, you would see depletions of cohesion of CTCF. And you see systematically changes that when you deplete loop extruder, so all the features disappear. When you deplete CTCF, features disappear, but there is still high density of contacts as expected, primarily because again, so there is no positional effect, but loops are still there. And when you put too much cohesion on DNA, you get all these nice grids of dots that appear at every between every pair of boundaries within a couple of megabases. So these are these beautiful experiments. And then, so this is like roughly 2017 and 18. And then the year 2018 and 19 brought us, and 2000, yeah, up to 2019, brought us beautiful experiments where single molecule experiments on condensance and cohesins. And what these experiments have shown is that cohesion, so that both class of molecules extrude loops. And again, the typical setup in these experiments is that there is a piece of DNA that is attached to a glass slide. So that's a movie about cohesion. There is a piece of DNA attached to the glass slide, so it's stretched in the flow. And once loop extruder is added, this loop extruder creates this loop. So let me pause now for questions, because I start seeing some questions popping up in the chat, and maybe just ask people to speak up and ask their questions. So let's pause for questions now. Just unmute yourself.
Yeah, please unmute yourself and ask the question, or if you want, I can read it.
Yeah, this is Dennis Tischer. Yeah. So have you simulated in a time-dependent way with a force velocity relationship that captures the experiments? I sort of missed the direct comparison. What sort of force velocity relationship do you have to get this?
That's a great question. That's something that we're working on right now. So we have not done that before, because in our simulations we assumed that loop extruder is infinitely powerful.
Which is probably not right, yeah.
Which is certainly not right. Which is certainly not right, because they do get stalled at about 1 piconewton force. So that's something that we're exploring right now. So can we reproduce this kind of loop force velocity curves that people have seen? One challenge is that even the stall force is not unambiguously measured. For example, this paper, let me see which one. So Case-Decker's paper gives a stall force of about 1 piconewton, and then this paper gives a stall force of 0.1 piconewton. So it's a bit hard to operate at this level of uncertainty, but that's something that we would love to understand better. So where the stall force comes from. So what is the? I think the biggest question now in this, or one of the biggest questions in this kind of SMC field, is what is the mechanism of extrusion? How do these tiny rings can extrude DNA? What is the mechanism of this motor activity? So this is still elusive, I would say. Kind of we're trying to do coarse-grained models of loop extrusion, so just to get the force velocity curves, but it's still hard to say what's the actual mechanism of extrusion. And the speeds are enormous. The speed is of the order of 1 kb per second. This is about 10 times faster than a polymerase, than an RNA polymerase. And it's comparable to bacterial DNA polymerases.
Hello, this is Golnaz Vahiri. I have a question about the dots and the example you showed. Can you elaborate a little bit more? So you think that doesn't happen frequently, or the dots don't correspond to transcription? So maybe I like to know a little bit more about maybe the link between expression and the dots.
Okay, the link between expression and the dots is again. So we don't really know what the link of what the link is to an extent. At this point, most of the visible, I will talk more about dots in a few slides, but most of the visible dots are dots between CTCFs. Now, what imaging people have told us is that, again, high C doesn't give you an absolute quantification of frequency. It doesn't tell you how frequent this event is. What high C can tell you is that the intensity of this dot is comparable to the frequency of interactions between two elements separated by some distance. And if the dot were of a contact frequency between two elements separated by 5 kb, I would say the dot is present there, like in the vast majority of cells. Typically, the dots are as strong as frequency of interactions between two elements about 50 kb apart, meaning that it's there, but it's not incredibly frequent. And then imaging works have shown that dots are present in about of the order of 10% of cells. And again, present in imaging means that two CTCFs are closer than 300 nanometers apart. It doesn't really mean that they're touching each other. And that's something that I'll discuss in a few slides, but at least it's less than 10%. So dots are not incredibly frequent in any kind of in a population average picture.
Good. Thank you.
Okay, let me proceed, and so we can discuss these things later. So the question is, what's the functional role of loop extrusion? Okay, so loop extrusion is there, we know. It's in vitro. We see evidence of this in vivo. know the turnover time of motors, and we still don't know their speeds in vivo. We can only get some estimates for how fast things are happening in vivo. But we don't know the function of this. So is this just to create TEDs and entertain us? Or so is there, what's the functional role of this? And I will, and again, I will argue that the functional role of extrusion is to actually mediate, promote functional interactions between distal genomic elements. But before going into this, one interesting thing about TEDs and the way people have been thinking about them is that TEDs are clearly functional in a sense that by removing a boundary, you can make regulatory elements in one TED talk to targets in the other TED, and that can lead to all sorts of disorders. So knowing this, you would assume that this insulation at the TED border is very strong, because you really don't want these elements to talk to that if it leads to an extra finger, like in this phenotype, so fused fingers, or it leads to cancer. The surprise, however, is that this insulation is actually very modest. That insulation created by boundaries is only about twofold. And then there are many evidence for relatively modest spatial insulation between TEDs that come from fantastic imaging works of Tom Mistelli, Xiaowei Zheng, Alistair Ting Wu, Eric Joyce, and others. So this insulation is not very strong. And the question is, if insulation is only twofold, but it's functionally very important. So this is again my comment about some of the illustrations that you see in the literature where people just white out these regions in the map and make you believe that there are no interactions between TEDs or these things. So clearly there are plenty of interactions between TEDs. So we should take these illustrations as cartoons. So there are lots of interactions across CTCF borders, and these borders are not very high. And the question is, how do TEDs work? So shall we panic and say that TEDs don't work? Or is there some underlying truth that's kind of hiding behind this high C maps? And I will suggest like 3 ideas that I think can help us understand what's going on better. Idea number 1 is very simple. Idea number 1 is that for a regulatory interaction, you really need transcription factors at one place talk to transcription factors or polymerase at another location. So in other words, you need small spatial separation between enhancers and promoters. And Hi-C does not measure this. On the contrary, Hi-C has a estimated contact radius of about 150 nanometers. And again, we estimated this indirectly. Others have kind of comparison between microscopy and Hi-C also shows gives similar numbers. So that basically means that instead of capturing what this element talks to over here is a finger of Adam. So this what we see in Hi-C are all the interactions with all the stuff in the cell at about 150 nanometers. So the specific interaction that is present or can be present is immersed into a sea of random stuff, or maybe structurally important things, but not really direct functional contacts. And the question is, what will change if I were to look at this kind of direct contacts? There is no method to probe for this direct contacts because neither imaging nor Hi-C family methods can approach this. So the best we can do is actually simulate this. So what we've done, we essentially run our polymer simulations at 1 nucleosome resolution. And then if I probe the simulations using Hi-C-like contact radius, I get Hi-C-like looking maps. But now the question is, what would happen if I were to probe this at 15 nanometers? And the answer is, I start seeing maps that are much sharper, much crispier. So small contact radius essentially increases the contrast of all of the features. And now the question is, maybe that would answer the question about the insulation. Maybe insulation is stronger for direct contacts or narrow range contacts than for broad range contacts. But before going into this, the question is, how can we support the notion that changing the contact radius or the spatial resolution of high C changes the contrast of features? So the evidence in support of this comes in the form of comparison to another method. So there is a new flavor of high C called micro C. So this is an MNase-based high C developed by Ole Rando's group at UMass Medical Center, again in collaboration with Job Decker's lab. What micro C does, it's essentially a high C protocol with MNase instead of restriction enzymes, other specific restriction enzymes, and with few other clever tricks and somewhat different ligation protocols, ligation and fixation protocols. So what we see in micro C comparing to high C is that the contact radius in micro C is about 50 nanometers, and indeed the features get crispier. So all these dots that may have been like maybe 7-fold become basically 20-fold. The contrast between the interior of the TAD and the neighboring region, or the stripes and the neighboring region, if it was like, again, 7-fold, it becomes about 20-fold. So things do get sharper at a smaller contact radius. And we see this also in simulations. So as I told you, the features get stronger at the smaller contact radius. Now the question is, what about insulation? Because that's where we all started. And if you look at insulation doesn't quite get much stronger. So it does not really resolve the problem of modest insulation. So insulation is about 2-fold in high C, and insulation remains about 2 to 3-fold in micro C, and it remains about 2 to 3-fold even in our simulated nano C. So we call this very narrow range contacts, so we call this nano C. It's a non-existent method at this moment. But still insulation is relatively weak. It can get stronger at places, but still not considerably stronger. So it does not really answer our question. So how can we live without strong insulation of enhancers? So enhancers can talk to other things, even at small contact radius. But what we also noticed is that so this is just to say, so insulation might be getting somewhat stronger for direct contacts compared to high C proximity, but this is not enough. So there is a second idea. And the second idea is that there is facilitation. And that's what is evident in high C maps. If you look at high C maps, you see these beautiful stripes that can extend for megabases in some of the very famous locus like the Sonic Hedgehog or the Mick locus. These are long stripes, and so how do they emerge? And I will explain this. So essentially, you can think about it in the following way. So imagine there is a gene that lives next to a barrier, and the barrier can be a single CTCF or a cluster of CTCFs. Now, loop extruders would be landing anywhere within this region, and then one of the loop extruders would extrude up to a barrier. It would stop here because it meets the barrier, but it would continue reeling DNA in. And as it reels DNA in, this boundary element would essentially be scanning all this region in search for its partner. And then when it meets its partner, so that's going to be the functional interaction. Remember, there is no other CTCF here. So it allows to scan. If I put the CTCF somewhere here, it would essentially interrupt the flame. And the lengths of the flame in this picture, I constantly switch between three terminologies that are kind of in my mind. We call them stripes, we call them lines, and we call them flames. So we'll try to stick to stripes, but if I start calling them lines or flames, so I'm still talking about the same features. So the notion is that loop extrusion can give you these features for free, essentially. You don't need to assume that there is targeted loading of cohesion at boundaries. And it would allow essentially every pair, it would allow this region to scan very extended genomic region. Again, on the language of high C maps, what's happening is essentially if you say, so that's the place where the loop extruder started, this would be the track of this loop extruder until it hits the boundary, and then it would continue along the boundary. So you can think about it this way. Now, this idea is not entirely new. The idea of stripes or lines, first, again, we noticed this in our first simulations in 2016, wrote a review about this at about the same time. Rafael Casellas and Eris Lieberman's lab published this paper, again, highlighting the importance of stripes, but arguing that they appear if there is loading next to the boundary. I don't think this is really necessary, but maybe it does happen in some cases. And then 2 years ago, it was a nice paper by Susanna Hajur's lab, demonstrating again the abundance of such stripes in real data. So what's new here is that what's going to happen if I combine this idea and the first one. So if I combine if I look now at the stripes, but using the narrow contact radius, I see enormous facilitation. If stripes in high C were somewhere like 4-fold, in micro C maybe 5 to 10-fold enrichment, as far as direct interactions between molecular complexes, stripes can go up to 40-fold. And this is 10 times bigger than any insulation that you can get. So it's a very, very strong facilitating effect of boundaries. And that, I think, is a cool mechanism to mediate regulatory interaction. So CTCF remaining an insulator is also an activator. And again, you can also look at micro C data, and you would see the stripes, and the stripes indeed get stronger when you compare them between high C and micro C. And we don't know, again, there is no experimental nano C. So this brings kind of a notion, which is almost like a metaphorical thing. We basically saying that extrusion boundaries play two roles. They're both insulators and facilitators. So if you, for example, measure the strengths of insulation and facilitation as a function of the contact radius, for small contact radii, insulation doesn't really get much stronger, but facilitation at stripes or facilitation at dots is really, really large, particularly at large contact radii. So high C is operating somewhere here. That's what we can see. That's a region where real molecular complexes might be forming. So things are much, much stronger there. And this duality is evident duality of CTCFs is evident if I start looking at CTCF clusters and I sort these clusters by their insulation. So they're sorted by insulation, and you see that this one is not really insulating. This one is weakly insulating. This one is insulating strong, and so on. At the same time, you see that even weakly insulating ones start generating some stripes. The stripes getting longer and stronger as the cluster becomes more and more insulating. And these stripes would presumably extend beyond this 0.5 megabase window that we're focusing on. So essentially, every boundary is both insulating and facilitating on average. On average, meaning that we see this in simulations and we see this in the data if we aggregate over many boundaries. But the moment we start looking at them individually, obviously things are much more complicated. So these are snapshots from the average pileups that I just showed you. So obviously on individual snapshots, things are incredibly complicated with many boundaries interacting with each other. And it's very hard to delineate their facilitating and insulating effects. But the take-home message is still that there might be some boundaries that might be facilitating, but not really insulating. And there might be that are insulating and not facilitating. But on average, these two things go together. So overall, I think the importance of this is that it suggests that CTCFs and play a role in facilitating interactions, and maybe global loop extrusion facilitates them. So let me add one more idea, and then I will take questions. And I'm actually basically at the end. And the last idea is that you can target these facilitate interactions to specific places. So this is our previous dock and scan diagram. So this is strong CTCF. That's a gene. This gene will be looking for its regulatory site along this region by dock and scan. But in addition to this, I'm going to place a weak boundary. And the notion is that once I place a weak boundary, this weak boundary would help this gene stay longer, interacting for longer with certain regulatory regions. In fact, you can swap regulatory regions and genes here in a sense that it might be a regulatory region that stays longer and interacts more with certain genes and skips others. It would interact with them, so they would be scanned through, but it wouldn't dwell there for longer. So by placing weak boundaries, you can kind of focus these interactions. In simulations, yeah, I will take questions in a second. In simulations, we see this very clearly. So if I start placing weak boundaries at particular places in the genome, they will become very visible in simulated nano-C. So they will start generating this stripes. Again, that's a relative to control. They will start generating this enrichments. If I in high C, in simulate high C, I'm not going to see them. They're just completely invisible, these local enrichments. Similarly, if I look at real data, comparing micro C and high C, in high C, this is just a stripe. In micro C, if I take a slice of this in micro C, this stripe is made of many peaks. What's also interesting is that basically every DHS site, every DNA hypersensitive site, or in other words, arguably every regulatory region in this neighborhood, serves as a weak pause site. So extrusion pauses at every DHS site. What's the mechanism of this? Is it steric or other specific interactions with cohesins? We don't know. But the take-home message is that DHS sites can serve as pause sites, thus focusing interactions with the dock. Not all of them do it equally strongly. Some are strong pause sites, and some are weak. But this gives another 3 to 5-fold enrichment of interactions. Yeah, and these are just more examples of this type. When you don't see these things in high C and do see them in micro C. These are just other more or less randomly picked loci. And again, you may be able to see them in high C in some cases, but in micro C, they're much more visible. Now the question is, what's at the docks? And what's at what elements are at the docks and what elements are along the stripes? Surprisingly, so I was expecting that there are going to be enhancers at the basis of the docks, and these enhancers are scanning long stretches of DNA looking for their targets. The answer is no. It looks like these are transcription start sites that are at the, so what I'm showing you is the density of various regulatory elements as annotated by Chrome HMM, Manolis Kellis's genome annotation platform that uses histone marks to assign function to every bin. So these are attack-seek peaks along the stripes from CTCF, from strong CTCF boundaries. I see a very large enrichment of transcription start sites. And then enhancers are not really enriched here. Enhancers are in yellowish. Enhancers are just broadly distributed over this kilobases of DNA. So this is up to 20 kb. So enhancers are somewhere there, but they're not really localized. So it looks like dock and scan largely operates, at least in these cells that we looked in. And these are ES cells. So these are genes that are docks, and they scan for the enhancers. Maybe in other cell types, the picture would be different. So I wouldn't be surprised by that. And that's the last slide with the information that I will show you. The question is this really that these pause sites are real? And are they really pause sites, not artifacts of micro-C? To answer this question, we compared interactions of a CTCF with what we call the scanning zone, essentially downstream from the CTCF in the direction of the CTCF motif versus upstream from CTCF. And it turned out that indeed, you see these enrichments at stripes only in the scanning region and not in the opposite direction. And with this, I will pause for questions, and the only slide that is actually left is the acknowledgement, but I will still pause here for questions. So there are several questions, so please unmute yourself. And ask your questions.
So I think, Sora, can you unmute, or do you want me to read your question?
Yes. So when we look into the stripe domain, we can find there are a lot of forward or reverse oriented CTCF. So I wonder what determines the boundary or the ends of the stripe domain?
When you ask the end of the stripe domain,
Yes.
this part,
The
or
dock.
the second one or the first?
Sorry?
Are you asking about the docking part, this one, or the other end, or this end?
I mean the anchor.
The anchor.
dock. Yeah.
Okay. The answer is some strong boundary. We don't really know what this strong boundary is. Presumably a large cluster of CTCF that is sufficiently strong to stop cohesion.
Yes.
In addition to this, what we now realize, I skipped through this, in the simulations that reproduce the stripes, what we also had to put in is the ability of the boundary not only to stop cohesion, but also to extend the lifetime of cohesion on DNA. So we turn this 10 minutes into 40 minutes, such that cohesion can travel this megabases of DNA in simulations. So if I go to any simulated one, yeah, for example, here. So in these simulations, what we had to do is to extend the lifetime of cohesions such that they can scan long genomic regions. So this is actually not so long. This is 300 kb. That may work without extension of the lifetime, but yeah, but to get this long stripes, megabase long stripes, you essentially need additional stabilization. So it's an ability of CTCF to stop cohesion, but also to prevent its unloading. What features of CTCF do this? We don't know. There was a nice crystal structure from Benjamin Rowland's group and Daniel Payne that argued that there is a specific motif of CTCF that binds to, that is recognized by cohesion, and then presumably this specific interactions by cohesion and CTCF is what stabilizes cohesion on DNA. They might be mutually stabilizing each other. And that gives us long stripes. Whether these are specific types of cohesion, SA1 versus SA2, whether other molecules need to be present or absent at the CTCF itself, like PDS5 that also can bind to CTCF, this is something to be understood.
Thank you.
So I think I read the next question. It says, are the stripes the result of slower extrusion? In the other words, is symmetrical extrusion faster than asymmetrical extrusion?
That's an excellent question. we don't know what's happening in vivo. We do know, okay, so to answer your question, yes, the stripes are a result of asymmetric extrusion that happens because cohesion is stalled at CTCF. What the stalling accelerates the other side of the motor, we don't know. There are measurements of cohesion. So some of the single molecule papers, and if I can rapidly jump there, some of the single molecule papers, this one, this one. So this paper observed asymmetric extrusion of condensans, but not cohesins. And it looked like, so nobody really has observed stalling of cohesin by CTCF in single molecules. So we don't know whether the speed would go higher. But it does look like generally one-sided extrusion is faster than two-sided. So I wouldn't be surprised if stalling a motor would accelerate its reeling speed. It's a great question. We don't know. But single molecule experiments will tell us.
I think the next question is, I think, is by Gerd. Gerd, do you mind?
Yeah, very nice. So I just wanted to get your views on the duality of the insulation versus promotion function of CTCF. And as we've discussed earlier a little bit, is that the position of the CTCF site with regard to the regulatory element seems to matter quite a lot. So for example, you know, when a regulatory element is flanked by a CTCF loop, it seems to contribute to insulating function. When an enhancer and a promoter are, you know, within a CTCF contained loop, then it facilitates those contacts. That would be in our South cycle study. So I wonder to what extent, you know, this can be modeled, but that's what our experimental data suggest.
That's something that I would love to model and look at with you, Gerd. It does look this way. For example, if you look at the diagram on the right, so strongly insulating CTCFs, you see that, for example, if I place two elements here, they would be insulated but not really facilitated at this distance. Mm-hmm. If I place it over here and over there, they would be facilitated. So chances are that there is some sort of distance dependent effect, but it may also depend on what is at the boundary. Like for weak boundaries, it wouldn't presumably be the case. So this is something that is certainly incredibly interesting to examine in simulation.
Why do you think then that if there's a CTCF loop that is encompassing an enhancer, for example, why can't the enhancer still not loop to its promoter? What's your explanation for that?
If CTCF, say it again. So
let's say you have an enhancer, and left and right of the enhancer, there's a CTCF site, and both of the CTCF sites are connected by a cohesin contact.
Right.
That enhancer is insulated. Why can't this? I mean, if enhancer promoter contacts, many of which, you know, occur completely independently of cohesin, perhaps presumably because they like each other, like-minded chromatin states. Yeah. Why can't those contacts form? Why can't it just loop over that CTCF loop?
I think they can, but it's the question of relative contact intensity. But the other question is whether enhancer and promoter interactions, if this were like a promoter, whether extrusion by itself can just simply break this affinity, override this affinity, like what it does for compartmental interactions, that the motor activity kind of breaks affinity-mediated interactions. Maybe that's how I would be thinking about this, but I don't. This is kind of more ATP-dependent process are simply stronger than these affinities. Why can't it carry this enhancer with it? I don't know. Maybe it can. It can just carry the whole thing over.
Thank you. More to discuss later.
Yeah.
I think the next question is Golnaz.
Yeah, I'm going to be very quick. You have a lot of other questions. So your simulation suggests that the stripes get stronger, but in our data, we have a lot of evidence of the stripes being very cell type specific. So can you just maybe quickly mention how, like, is this information only encoded in DNA, considering that it was coming out of the simulation? Just I'm curious about that.
Simulations are incredibly, simulations have a boundary. In simulations, we don't have CTCF explicitly. We have a boundary, and the boundary has certain properties. At this point, so the boundary has two properties. It can stop, and it can give a bonus in the lifetime. What molecular processes contribute to ability of a boundary to stop and to ability of a boundary to give a bonus in the lifetime, we don't know. So for example, in different cell types, you can either lose CTCFs there. You may still have CTCFs, but the CTCFs would lose their PDS5, or have PDS5A replaced with PDS5B. Kind of, you can imagine different molecular process at CTCFs themselves that would make them kind of given this bonus in the lifetime or not given the bonus in the lifetime, for example, or stopping cohesion differently. So it's yeah. So these things can very well be cell type specific, because present kind of what's encoded in the DNA is the ability to establish a boundary there. But it doesn't necessarily mean that there's going to be a boundary there. Nice examples of disappearing and appearing boundaries are the works on protocadherin locus. So in the protocadherin locus, so boundaries are initially not established before the choice has been made about which specific exon is used. So it's an alternative exon usage. And then expression of a small regulatory RNA stops cohesion. And if cohesion stops there, there are many alternative places. Then a DNA demethylating enzyme is recruited that demethylates this region, allowing CTCF to bind. After CTCF is bound, this solidifies the loop, and the decision about the exon choice and the type of protocadherin has been made. So that's a beautiful paper, cell paper from last year. The first author is Daniel, and I cannot recall his name. I'm sorry.
Thank you so much.
So maybe I skip my question, and then we go to Eric. Eric, do you want to unmute?
Yeah, thanks, Babak. Hi, Leonid. Question is, how does the position of cohesin loading impact your model? Like, I heard one say that it didn't really matter, but I guess in your figure, the enhancers are not preferentially pause sites along a stripe. Wouldn't your models predict then that they are preferentially cohesin loading sites to begin scanning?
So in the, I don't know what's the right, okay. So in this diagram, so we actually never assume that there is specific cohesin loading. So everywhere we just assume that cohesin is uniformly likely to load anywhere and unload everywhere. So in this sense, you don't really need to get cohesin loading sites specifically at any places to explain what's going on. So completely uniform loading of cohesin can generate you, I don't know, this kind of maps, for example. So in other words, cohesin can load anywhere. I don't know where to draw here. It would start forming progressively larger loop until it hits the boundary, and then it continues forming the loop. So it doesn't need to load in specific places, and it doesn't need to unload at specific places. To generate an asymmetry, what you need is, for example, this CTCF presumably needs to generate a stronger stripe on the right. It would presumably need to stabilize cohesions approaching from one side than the other. So the only asymmetry in the system are positions of CTCFs and their orientation. So otherwise, you don't need for kind of specific loading, but it doesn't mean that there is no specific loading. It means that a simple model like this doesn't really require it. So the moment you go into real data, things may change. So in a sense, I don't know, something happened to my computer.
All right, thanks.
Like to explain what's happening in reality, maps like this, I bet you may need lots of other things to happen.
So we have 1 more minute left. So I think we can, if the speaker doesn't mind, maybe we have 1 more question.
No, absolutely. I can still, I just need to acknowledge people who did the work.
Sure.
And give some, okay, let me give a summary. Okay, the take home message that I was trying to deliver is that maybe we should replace the picture of isolated neighborhoods, kind of separated by high walls, with a picture where interactions can be directed to specific positions, and where in fact installation might be less important than facilitation. It might be very local, and facilitation can be directed. And we're yet to learn the rules of the road. So it's essentially some sort of communication device between different places in the genome that stop or directionally stop extruders. And the people who did the work, this a kind of fantastic collaboration with groups of Ole Rando and Job Decker on micro-C, and the main players in this work are Max Imakaev, who is Max Imakaev, Nizar Abdenur, Jeff Udenberg, and Samir Abraham. And sort of Jeff is now starting as a faculty at the University of Southern California. Nizar is still with us and will be on the faculty on the job market for position in data analysis. So if you're interested, so he will be going into the job market soon. And Max is now continuing his career in BioBot startup that is tracking COVID viruses in sewage. It's a really cool startup, so I'm very happy for him. So with this, I will take remaining questions. And thank you.
So Mustafa, do you want to ask your question or?
Yeah, sure. Hi, Leonid. A question related to 2 points you raised. First, that you said that the twofold insulation is relatively weak. And second, that enhancer and promoters need to be in close contact with each other. Mm-hmm. So it's sort of a question related to both of them. So first, given that we know this importance of these weak protein DNA interactions in creating sort of fluctuations in the local protein concentrations, do you think this twofold enrichment is enough to drive the creation of these local microenvironments of protein? And within these local microenvironments, then can enhancers and promoters interact with each other without coming into these close contacts? So I just wanted to pick your brain on this idea.
It's a great question. And I've been thinking about this, and there are many pictures where people are basically saying, so there is an enhancer and a promoter, and they are surrounded by some sort of a condensate. And then within the condensate, there is like some sort of a spooky action at a distance. Somehow they interact through the condensate without coming in direct contact. It's still hard for me to believe in this. I can tell you. So I can see that there is space for phase separation between heterochromatin and euchromatin. Euchromatin might be forming these condensates, but it's hard for me to imagine molecular complexes interacting with each other specifically without forming a direct contact. Something should be happening within this condensate and these condensates are got to be large, so 150 nanometers. And people constantly see them in microscopy, but it's hard for me to buy that this is the actual kind of interaction. Your second question was about twofold. I can certainly see a situation where the actual change in gene expression as a function of the contact frequency, if this is like contact frequency, and this is transcription, that this function is highly nonlinear. This I can certainly imagine, and there are beautiful works from Luca Giorgetti that he speaks about that hasn't appeared on the bioRxiv, and from Alistair Bottiger, where that is on bioRxiv, where they basically say there is a small change in the contact frequency, and that leads to a large change in transcription. And I think they're both right. What I'm arguing is that this is part of the picture. In reality, maybe contact frequency can also has a much bigger dynamic range than what we're seeing in high C. It's more than twofold. It might be like 10, 20-fold. And it's very localized. And yes, transcription then would respond the way transcription responds, depending on transcription factors and stuff like that. That I'm totally on board with. Yeah.
Cool. Thank you.
Okay. So please join me to thank Dr. Mirny again. I hope you all enjoyed the talk, and good luck with the rest of your visit.
Thank you.
Bye.